Okay, so I guess we have to start now. So, welcome to the class, everyone. Very good evening. It's going to be a new topic that is a biomolecule chapter. Okay, so let's continue. This is going to be the first session for this chapter. So, let's just start. Okay, so let's continue. First of all, like as the name of the chapter is the biomolecule. So, what is the meaning of this word biomolecule? Okay, in the lower classes, I guess, very good evening, Ambika. I guess you heard about that as the nutrition in our body, right? Vitamin, or you can say protein, or you can say carbohydrate, fat molecule. There's just so many things, right? So same. Uh, that is, this is not as a simple a nutrition kind of thing connected with the biomolecule, yes. So what is biomolecule? Let's just discuss more about this. That what is biomolecule? The living system are made up of various complex biomolecule like carbohydrate, protein, nucleic acid, lipid. If you don't know about the nucleic acid, maybe you already heard about the DNA. So the main monomer of the DNA is the nucleic acid. But are all these things, don't worry, we are going to cover all these things one by one. And then biomolecule interact with each other and constitute a molecular logic of a life process. For example, let me tell you one thing. Like let's just suppose we want to digest the food, right? So how the complete digestion is there? First of all, you to eat, right? And then that food transfer in your body, and then if fat is there, it is going to convert into the fatty acid. If protein is there, it's going to be converted into the amino acid. If carbohydrate is there, so it's going to convert in the glycogen and glucose part here. There. So means that we have to break them into the in the very small point, and they transfer to their particular like place, like there where we are going to consume this. So like this is the meaning that all process interact. For example, if in your body that is a deficiency of a protein, so our body is not working properly. If carbohydrate still not working properly, if there's a even little bit deficiency of a vitamin, so yes, we are facing some difficulty, right? So means what is the point that all these things, all the process, all these molecule combine and then up essential for a life process. Okay, so it means biomolecule play a very much important role for our living purpose. Okay, food like everything is very important. Okay, then now let's talk about the biochemistry. So, generally, maybe you heard about this that biochemistry topic. So, what is biochemistry? Biochemistry is that branch of chemistry where we are dealing with biology. Okay, as I already told you earlier, this topic is somehow connected with the biological classes, be like breathing and glucose and carbohydrate. Okay, so little bit biology concept. So bio means bio and chemistry for chemistry. When bio and chemistry combine and then become a new branch, it's known as a biochemistry. For example, if let's suppose I have to go with the genetics of a biology, okay, in biology there is a chapter that is known as a genetics. So what is the meaning of the genetics? Like for example, heredity concept, how to transfer the gene, everything, DNA, RNA, mitochondria, everything, so many things is there. But see, in chemistry we are not going to discuss about that. Is okay, DNA is the thing that transfer from one generation to another generation according to the chemistry point of view. Like what is DNA, what is the chemical structure of the DNA, okay, so how it is made up of. So that this is simple, that is a biochemistry means the biology deal with some chemical reaction. That part we are going to cover here. So, the parachute of the knowledge of what is going on chemically within a living organism is known as a biochemistry. Okay, so this is a simple meaning. Now, so like in this chapter, which topic basically we are going to cover? So, in the biomolecule part, we are going to cover about carbohydrate. Okay, so you know what is carbohydrate? Main point, you know, right? Protein, enzyme, vitamins, nucleic acid, hormones. So these things we are going to cover one by one. So in very much detail, no, it is a part of a biology. We have to cover only the chemical point of view. Okay. So let's discuss about the first topic of discussion is going to be the carbohydrate. So in the carbohydrate, we are going to cover these things one by one. So what is the point here? Look at this. First of all, introduction part, like what is carbohydrate, and then what preparation part, importance, classification, structure. So all these things we are going to cover one by one. Okay. Anyone, do you know that what do you mean by this word carbohydrate? Maybe in 10th class or maybe in 8th class, maybe somehow you deal with this word carbohydrate. So do you know what is the like logical or concept? What do you mean by this word carbohydrate? Okay, so If let's suppose I have to apply my logic means ma'am carb be like carbon have to be there. Hydrate means water have to be there. So it may be the combination of carbon plus water, right? Maybe it can be possible. So yes, like this in the previous time, there's just so many definition. Okay, there's just so many definition, but again that somehow the definition failed, and then we are end up with the new definition. So first of all, let me tell you a general definition. Okay, like that old definition you can say. Okay, so listen my point that in the old time, I guess you already everyone you know the formula for the glucose, right? So what is the formula for the glucose if I to write there? So C6H12O6, right? Everyone you know this formula, not an issue. Okay. So in this case, have you ever observed the point that is actually what is it? It's C6 and then water. Okay? It's water and then it's 6 time. Okay, that's it. Actually, this is the case everywhere. That the combination of carbon plus water is actually the carbohydrate. Okay, so then we like we can cover more formula. You know the formula for sucrose? Yes, please. Anyone? Do you know the formula for sucrose? Come on, you already covered sucrose type of question in which chapter? Solution chapter, right? C12. Yes, very good. Okay, so what is the formula here? Let me just write. That is the C12 H22 and O11. So again, if you're observing something that it's nothing, it's actually the water. And let me just write 11 time here. Okay. So like then at that point of time, scientist was very happy. Okay, we search the definition of a carbon. Then let's discuss about a formula that is COOH and COH. Wait. Okay, let's just cover this point later in the definition part. Let me just back again to our doll definition. So what we're discussing now, definition part here. So let's discuss more about the definition. So according to the old definition, I'm repeating this definition is not going to applicable everywhere. There's some limitation for this definition. Just try to understand. So what is carbohydrate? That is carbon plus water. That when carbon combine with water. Okay, first point. The first definition. Second definition, they are the hydrate of carbon. Just same point, that is if this is C6H2O and like 12 formula. So actually what is it? It's like a carbon combined with water. So it can be hydrate of carbon. Okay. And the third point, that is most of the time this is general formula. Like if you're observing C6H2O and there's some Y have to be there. Okay, we just record the sucrose formula as well. That is 2 and then water and then 11 time there, right? So it's a general formula. It's a general formula, but you know everything has a limitation, right? Let's cover that point as well. So first of all, observe this that in the example part, this is a glucose formula, and this is also can be written in this form. And according to the old definition, it can completely fit in this formula, in the given formula, in the old definition. And first into the general formula, this. Okay, like where x and y can be same. I'm not saying it is same. Okay, see, we are using different. X and y is a different symbol. Okay, let's just say this discuss about the previous formula that is a C12 and H2 sucrose formula I'm mentioning here. So see this is 12 and this is 11, right? So it may or may not be same. That's why we are using different variable x and y. So but in glucose both are the same. Okay, this is a simple sorted point. Now let's see some important part here. Okay, this fit definition fit everywhere, but the problem is what? That problem is very simple. If we are discussing about C3COH is there, so everyone you know it's acetic acid. I can write this is spelled like C2 and that is H4 and O2 there. Okay, so I can write this formula C2 H2O and 2 time. Okay, so what was the problem with the previous definition that if you have to go with acetic acid, so it resemble with this given formula that Y is there. But everyone we know acetic acid is not a glucose. Acetic acid is a acid, it's not a carbohydrate. So the fail, it means the all definition fail. Yes. So acetic acid fit into the general formula, but it is not a carbohydrate. So problem there. Second of what, that most of the time I just told you that every time water have to be there. But again, that is the raminose is a sugar. Okay, so what is the formula for the raminose? Okay, it is not visible here. So it's this is the formula for the raminose. That is a C6H12 and O5 is there. Okay, yeah, let me just check the formula. Okay, fine. So look at this. That in this case, what is the problem again? Look at this. It is not be like this. Okay, no, it's not fit. So again, that a sugar, that raminose is a sugar, and still it is not behaving like a general formula. Okay, so it means fail. So 2 point that why the old definition failed. Acetic acid was the limitation, and raminose is also the limitation. Okay, this is the only chapter where limitation also very much important. Why the definition failed. Okay, so that's it. This is all for the raminose. Now scientists be like, let's why not we can give you a particular definition, a fixed definition that applicable everywhere without any limitation. Okay? So now, according to the new definition, it is the definition given by the scientists. So what was the definition? That first of all, carbohydrate. Okay, let's break. Okay? Just try to understand. I will give you the complete definition first. So what is carbohydrate? Carbohydrate are ketones. Okay, ketose, or that is aldose. So what is the meaning of this word ketose or aldose? Means a carbohydrate have to be at least one aldehyde group or ketone. The point number one. First, this is the case. Or maybe it is not present in a general formula, so on hydrolysis. Either on hydrolysis that it give the same, that is a ketose or aldose. Okay, means what is the point? Basically, a ketone or aldehyde group, okay, should be present in carbohydrate. If they are not present in initial structure, but if you to hydrolyze them, maybe then they are generated aldehyde in a ketone group. Okay, so this is a fixed definition, no limitation. It can be applicable with every kind of formula. Okay, so let's discuss about the new definition part then. Then according to this new formula, what is carbohydrate? Carbohydrate are usually defined as polyhydroxyaldehyde. Okay, why the word is poly? Poly be like more than one hydro OH can be there. Single can be there and more than one can be there. Okay, aldehyde or ketone or the substance that hydrolysis to yield. Okay, maybe initially present. If not present, add them into the water, and after once the water is added, okay, maybe we are ending with this. So example be like glucose, fructose. We discussed this thing so many time in the that is the inversion of cane sugar, sucrose. Remember that when you to add the sucrose in the water, we are getting a two kind of monosaccharide there, that is the glucose and the fructose. Okay, the first point. And among all the carbohydrate are chiral and optically active. This is the point. Every carbohydrate have to be optically active. Optically active, remember that how to check the optical activity. Okay, let me quickly give you the brief that how to check it the optical activity. So first of all, we are going to check it for the chiral carbon. Okay, chiral carbon. And after this, then we are going to check it for the plane of symmetry or the center of symmetry, right? If anything present, maybe POS or COS. So if they are present, okay, so the given thing is optically inactive. Given molecule optically inactive. I said inactive. If it is absent, so it's become optically active. Okay, this is the point. Optically inactive. If POS and COS present, optically active if it is absent. Okay, can you just tell me actually, right now I'm not remembering the thing. We finish the stereochemistry or this is left? I guess no, right? Stereochemistry topic still left. Okay, just let me know if you know. Okay then. So this is all for the case. So let's just back again. At least we know this thing from isomer part, that is coordination compound as well. Okay, if you don't know, so it means that every carbohydrate have to be chiral except one, that is 1,3 dihydroxypropanone. Okay, so what does this 1 3 dihydroxy propanone. So let me just write the structure. 1 carbon, 2 carbon, 3 carbon. Okay, you can even check as well. That is first carbon, second carbon, third carbon. So first carbon, third carbon, that is a hydroxy. So it's be like 1,3 dihydroxy. 1,3 dihydroxy, and it's a propanone. And then some will be like, ma'am, isn't it that is it's going to be the 2 propanone? No, propane is always on the second carbon. Propane can't be on the first carbon, otherwise it become aldehyde there. Okay. So in this case, if you're observing something that in this there is a plane of symmetry present, okay? See this yellow plane here. So it is reflecting the thing. Okay, so if you're observing this, that is this OH reflecting, okay, hydrogen reflecting, and this is also reflecting. So what is the issue with this glucose? That is in this case the plane of symmetry present. Okay, let me just write that is what is this POS, that plane of symmetry. Okay, plane of symmetry present. So just because of the plane of symmetry present here, that is it is optically inactive. Okay, so like exception you have to note it. Let me just back again. All carbohydrate are optically active except this 1,3 dihydroxypropanone. Okay, this is all for the like introduction. What is carbohydrate? Any carbohydrate usually define the polyhydroxyaldehyde or ketone, otherwise, or maybe that on hydrolysis they yield this thing. Okay, now let's discuss more about the carbohydrate classification of the carbohydrate. We have to classify the carbohydrate into three types. Okay. So my question to you, what do you think that sugar is always sweet? Yes, please. If I use this word sugar, so sugar is always sweet. What do you think? So if you have to apply a normal mind, right, I can say yes, sweet. Okay. Next question. Carbohydrate is always sweet. So listen, carbohydrate may or may not be there. Okay. So if I'm referring the word sugar, so actually, table that is in table we are using which sugar? Sucrose, right? Cane sugar. So anyway, let's discuss about this taste part. So how about the taste, like sweet or not, and the behavior on hydrolysis, like ketone resulting, or like what about the molecule one, 2, 3, and 4 up to so on, reducing and not reducing sugar. Okay, if we are using this word reducing, so I'm referring here that is a Tollern test and Fehling test. Be noted why I'm mentioning this point here, that is a Tollern and Fehling test, because hey, that is what is carbohydrate there, that is carbohydrate is a ketone or aldehyde. And we know Tollern test and Fehling test is given positive by the ketone or aldehydes. If you don't know what is Fehling test and Tollern test, please revise again. That is a carboxylic acid chapter. It's play a very much important part here. Anyway, let's discuss more about the taste. Okay. Oh, wait a minute. Okay. Okay. So let's move further. So we're discussing about the taste, right? So on the basis of the taste, we have to classify the carbohydrate into the two part. That is, one is sugar and one is a non-sugar. Okay. So as I already told you, sugar is sweet and non-sugar is not or not may or may not be sweet here. So let's see that is what is sugar. So sugar is a carbohydrate. Sugar is a carbohydrate that are sweet in taste. And what is non-sugar? Non-sugar is lot that is don't have any sweetness. Anyone, do you know a carbohydrate which is not sweet in taste? Yes, please. Do you know any sugar which is not sweet? Sorry, carbohydrate which is not sweet. You know cellulose? You know cellulose? I don't know. Maybe you know. Do you know tree in the stem of a tree that we are having a bark that is brown in color. So actually, yes. So cellulose play a very much important part here. Even cellulose used to form so many water repellent thing. If you know sanitary napkin, so there we also using the cellulose, right? So of course you know. So now let me give the example here. So in this case, how about the example? So in this case, the example is milk sugar or cane sugar. And now, what is milk sugar? Okay, it's important, okay? Everything is very much important because you know this is a little bit biology. So every name has its own importance. Yes, please. What is the meaning of the milk sugar? Cellulose and starch. Yes, please. What is milk sugar? You know lactose. For the 6 month for a baby, it is recommended the mother milk only. Why? Because maximum lactose present in a mother milk, you know. So what was that? Milk sugar. Okay. So be noted lactose. Even it can be present in any milk, but in maximum present in a mother milk. Okay. So next one is cane sugar. So cane sugar that we are using in our home, that is sucrose. Sugar cane. Why? Because it is abstracted from the cane sugar or the sugar cane. Okay. Wait, I'm changing this. It's a starch. Okay, two thing. Okay, now my next question to you. Can you just tell me that how a human being stored glucose in a body? In which form? Options are starch, glucose, glycogen. How the food, how the energy stored in our body. Okay, don't say I'm ATP, okay? Don't say I'm in mitochondria. I'm not asking out in the cell point of view. You know the smallest form of food in our body. Okay, let me change my question. Uh, Sometime we are fasting, right? And on that day we are not eating anything, but still we are able to manage our body function. So what was the reason? Hmm? Glycogen. Okay, so the stored sugar in our body is the glycogen. Yes, and the stored sugar in the plant is the starch. Be noted. You know, just because of that, the starch is the main point in the plant that or the that which is the herbivorous. The large intestine is large in size, but in our body, small intestine is large because the smallest form is the glycogen. 10th class topic, you know. I don't know the Japanese, but I guess you know little bit about this. Anyway, that's it. Very good. So glycogen or the that starch here. So that's it. This is all for the classification part on the basis of the taste there. Now let's discuss of the behavior of a hydrolysis. Behavior of a hydrolysis means and what is this monosaccharide, polysaccharide, oligosaccharide? And wait, I miss a point here. Do you know that is the sugar which is sweet in taste is also known as saccharide? Okay, it's a saccharide. Saccharide. Okay, what you want, you can call it. Okay. So what does it? Saccharide, sweet. When sugar is sweet, is known as a saccharide. So yes, let's more discuss about the carbohydrate on the basis of a hydrolysis. Okay, let me give you a kind of brief of the previous classes. You know that it is a very common reaction if you have to add a sucrose into the water. Okay, so what we are end up there, that is a glucose and a fructose. Okay, so you know what is it? It is a bi, okay, or you can say that is a disaccharide. That when you to add the starch into the water, so we're getting two molecules. That is molecule number one is the glucose, molecule number two is the fructose. So glucose is the smallest unit. Okay, you can say it can't be further that is break into some other molecule there. So glucose is a monosaccharide. Why the mono? Mono means single unit. It is not further hydrolyzed, and fructose is also the mono there. But see, this is the sucrose. Sucrose break into two molecules, so it's become by, if three molecule tri, four molecule that is a tetra, and up to so on. And this is how we have to classify the sucrose on the basis of the number of molecule given on hydrolysis in the water. Okay, so let's just back again here. That is what is monosaccharides, that no hydrolysis. They can't give even a single molecule on the hydrolysis, or you can say not hydrolyze further. What is oligosaccharide? That is, it's a particular number that you can say 10 to, okay. So you can say in this case 10 to 10 molecule, 2 to 10 molecule, I'm sorry. And if it is greater than 10, so it's become poly. You can say so many number. Poly means n number. Okay, so this is the meaning. Okay, then let's go. Let's discuss more about this. So what is this monosaccharide? So what is monosaccharide? Monosaccharide can be classified basis on the functional group and a number of carbon atom present in a molecule. It is, first of all, function group, as I already told you, maybe aldehyde, maybe ketone. So if aldehyde present, we used to use this word, that is aldose. Aldose means aldehyde, the sugar which contain aldehyde, or ketose means the sugar which contain ketone as a functional group. So for example, monosaccharide containing aldehyde group and a three carbon atom is aldotriose. Aldo means aldehyde. Okay, triose means that is in this case total three carbon present, three carbon present. See, if ketone combined with more four carbon, okay, so it's become keto tetrose. Tetra, tetra for four. So it's become keto tetrose. Okay, so this is how the nomenclature for the glucose you can say, monosaccharide. Okay, then now oligosaccharide. So what is the meaning of this word oligo? A very few amount. Okay, you know that how lengthy the protein, vitamin, and carbohydrate. There is a million or billion or trillion thing in our body, right? So if we are using this word oligo means few. Few means 10 to 2 to 10 is a few number here as compared to the million part. So what is oligo? Few. Saccharide means sugar. So the carbohydrate that yield 2 to 10 monosaccharide unit simple carbohydrate on hydrolysis. What is the simplest form? Glucose, fructose, lactose. Okay. So glucose, fructose are the smallest form here. Okay, mannose as well. So let's see that is what is disaccharide then. So it's further classified as di, tri. So what is disaccharide? So two monosaccharide unit are obtained on hydrolysis. Sucrose give glucose, fructose. Everyone, I guess you know, at least you have to know this. We know from this from the solution chapter so many times we discuss about that what is the one two factor for the sucrose, or if you to add into the water hydrolysis there or not, we discuss so many times, right? Maltose. Okay, formula similar because they are the isomer of one another. So sucrose and maltose, the formula be like 12 and H22. Even this is the same formula. But what is the difference? How to differentiate between sucrose, maltose, and lactose? Okay, so I know that what is the problem with the biomolecule chapter. You have to remember so many things. But everything is important. So now all three things should be in everyone mind, clear, not an issue, easy and important, and already asked so many times that if you to add sucrose into the water, glucose plus fructose, maltose, glucose, lactose, galactose, and glucose. I know you can remember the rhyme. Like I used to remember lactose means galactose is the extra point. Glucose, glucose are common. Glucose, glucose, glucose. And sucrose, fructose. Maltose, extra glucose. Lactose, galactose. So this is how that important. It's really important part. Now let's see the next one. That is polysaccharide. So what is the meaning of this word poly? Poly means many. Saccharide means sugar. So the carbohydrate, sorry, generally, they are not sweet in taste or has known as a non-sugar. So be noted what is non-sugar, the sugar which is not sweet in taste. Most of the time polysaccharide as we discussed, starch, glycogen, gum or cellulose are the known sugar, not sweet in taste. So the carbohydrate that yield more than one unit on hydrolysis we is known as a polysaccharide, be like starch, glycogen, and gum. Let's brief this all the thing what is mono, what is poly, and all these things. So observe this slide, everyone. Monosaccharide when the number of product form only one, glucose, fructose, and galactose. Oligo means 2 to 10 units, sucrose, maltose, and lactose. Okay, polysaccharide means more than 10 unit, the starch, glucose, glycogen, and you have to add gum as well. You know the gum, okay? I hope you already know the meaning of this word gum. Okay, maybe you don't know about this, but in the old time, again on a stem of the tree, okay, there is a sticky thing have to be there. And we used to collect that, and that is known as a gum. Okay, maybe you don't know because you are the new generation child. Okay, anyway, so that's it. Now let's see the third classification, reducing and non-reducing sugar. So what is reducing sugar? As the name suggests, that is something is connected with the reduction here, okay? So quickly recall the Tolan test. So what is the Tolan test? You will be having a some kind of CO group, right? Okay. Next question to you. Tolan test is given positive by aldehyde or ketone. Yes, generally you're right. Yes, please. Tolerant test is given positive. I guess today the screen is out of the wait. At least you have to mention, okay? Because I don't know about this. I guess it's covering now. Aldehyde. So tolerant test is not given positive by the ketone, you mean to say. Arul? I'm just checking the previous knowledge. So if you know, let me know. Okay. So very good. It is going to be aldehyde, right? So but again, there's a twist in a story. I will tell you later that here the tolerant test is also given positive by the ketose as well. It means the alcohol which containing ketone. We will cover that point as well. Wait. Okay, so this is aldehyde, and we are using silver mirror. We are using a silver there, and silver is present in plus one oxidation state. It is going to convert in the zero oxidation state. Silver mirror appear, and then it is going to convert in the kind of acid there. So it's oxidation, it's simply reduction there, okay? So why the topic name is reducing sugar, a sugar which reduce the tolerant reagent, right? So yes, again on the basis of the reduction, we have to classify the sugar into the two part, that is reducing sugar. So the carbohydrate that reducing the Fehling solution and the tolerant reagent is known as a reducing sugar. Or that is example fructose, glucose, maltose, lactose, etc. Carbohydrate that do not reduce the tolerant test or the Fehling solution is known as a non-reducing sugar. Again, it is a very important example or most commonly asked PYQ, which of the following is a non-reducing sugar? Sucrose is a non-reducing sugar. Okay, what is your doubt, Sid? If you are in the group, just post the doubt there, and then here I am explaining the thing, okay? You can ask, please, okay? Just do this. Let's go again. If you are having a problem with the ongoing thing, so just let me know. Okay, so B noted sucrose. So all monosaccharide where ketose and aldose are reducing sugar. But ma'am, you told us that aldehyde is only give the tolerant test positive. Then how it is possible with the ketone? Okay, so anyone, can you just tell me a one limitation of the tolerant test? Kind of ketone also give the test positive. Anyone remember? Unsaturated solution. If you are saying sugar, so sugar is a polar compound, so it can't be. But you know, in a laboratory, we can use the benzene as well. Okay, because the problem be like it's a carbo, it's a actually organic compound as well. But if you have to go with lactose or like, so sugar is not very much soluble in unsaturated solution. I guess unsaturated means you want to say organic compound, right? Unsaturated, can you just be particular? What do you mean by unsaturated solution here? Because unsaturated be like, benzene can be there. Yes, please tell the anyone. Can you just tell me the limitation of the tolerant test? Some kind of ketone also give the test positive. Okay, because the answer lie here. Like how ketone give the test positive. If you are asking general one, yes, it is a yes. Okay? Yes, it is soluble. Even not only fructose, even glucose as well. Okay, Sid? Okay, so let me quickly give you a brief if you remember. Okay, that is alpha hydroxyketone if you remember. Mm. For example, I said alpha, beta, I'm sorry. Okay, so actually how it's this is how the glucose structure look like. Just try to understand. Okay, so in this case, what is happening there? You know, after the tautomerism, this CO and OH switch the position. Okay, so let me tell you how. After tautomerism thing look like that it become OH and it become aldehyde. Okay, so although in the very first it is a ketone, but again, if you're observing that with respect to this is alpha, so it's alpha hydroxyketone. So alpha position if OH group is connected and terminal OH. Okay, it's not like you have to consider the center, terminal have to be there and it is going to convert. Maybe it is not very clear for you right now. Let me first draw the structure. Okay, yes, very good, Arul. Alpha hydroxyketone. And then we are able to find out the answer, like actually what I want to explain to you. Okay, just wait for the structure of the fructose, then we are going to cover this topic again. Okay, so that's it. So this is a question for you. It's a PYQ. Answer this question. It's an easy question now. Which of the following is a non-reducing sugar? Okay. Which of the following is a non-reducing sugar? Yes, very good. Okay, simple. Option A is the correct here. Okay, we discuss sucrose is a non-reducing sugar. So that's it. This is all for the case. Now let's see the preparation part. So we just covered that is a classification part or a definition part or yes, that's it. Now let's more discuss more about the preparation part. So we are going to discuss about the preparation of glucose. One preparation we know, that is use the starch. add into the water, we are getting glucose plus fructose, right? And we have to use acid and water as well. If you know pseudo first order reaction from the chemical kinetics, this is the same reaction. Sucrose plus water plus little bit amount of acid and boil that, and we'll end up with the sucrose plus glucose. This is also known as an inversion of cane sugar. Why it is known as the inversion of cane sugar? Every point we are going to cover one by one. Okay. Or the second point, that is you can use starch or you can use the cellulose, and it is really much important here. What is the formula? C6H10, C6H10 and O5. This is the general formula for the polysaccharide, starch or cellulose. You can use both the cases. And then diluted HCl we're adding. If you are no, if you don't know the temperature in a paper, and if let's suppose you miss the pressure as well, so never mention the wrong thing in a paper. Just use diluted HCl and okay, it will work. Okay, so we are getting glucose. So two method, by starch adding into acid, or by the sucrose adding into the water, simple hydrolysis there. Okay, this is all for the preparation of the glucose. Now let's discuss a very much important point, structure. This is the now the real story begin. And okay, you finish all the thing. After you can say hopefully you'll get a very good colleges. Otherwise this glucose structure, okay, follow you everywhere throughout the graduation. It's a tricky formula, or you can say bad structure. I really don't like this structure. So let's see what is carbohydrate or the glucose formula. So first of all, in glucose we are having six carbons. So just try to draw the structure again and again, two to three times, and it will be easy then. So simple. First of all, one carbon, two carbon, three carbon, four carbon, five carbon, six carbon. Done. Count 1 2 3 5 6. It's a straight chain. Okay, and then it is aldose sugar. So first of all it become a aldehyde, carbon number one. Let me just write the number of carbon as well, right? So this is my first carbon, second, third, fourth, fifth and sixth. Okay, so now you have to just count. So in this case, that is OH this side. Okay, only at third carbon OH this side, and then OH, and then OH, and then OH. Okay, this OH position is really much important when we are drawing the fissure projection, all this thing. Okay, I'm repeating one more time. Okay, let me highlight the important point here, that every time OH this side, this side, this side, this side, but only this is the third carbon where the OH on the like you can say left hand side. Okay, so left and right, if you are having issue with the left and right, okay, so just use any trick. But like this is a left side here and this is a right side here. Why I'm mentioning this? Because when we're discussing about the Allen dextrorotatory, so it's important here. Okay, of course. Wait, Ambika, which part is not clear to you? Again, it's out of the screen, right? Wait, okay. I guess it's okay now. So which part is not clear, Ambika? Where's the slide? This part, Ambika, just please confirm. This is not clear? Okay, preparation of glucose. So listen then again. First of all, this is the key method number one. If you are using sucrose and if you have to add into the water, so it's going to break. Like it's a C12, H22 and O11. So 50-50, half of portion. So 12 becomes 6, 22 become 11 here, sorry, 12 here, and then oxygen become 6 here. Okay, it's a like water addition there. You have to add water as well. So this is how you can count the carbon here. 22 from here, 2 from here, so it's become 12 total, 24. Wait, I'm explaining on the next slide. So C12, okay. So here we are using water as well. Okay, so you add this like C12H22 and water. What we are having 12 carbon now, total hydrogen, 2 from this, 2 from this, so it's become 24, and 11 from this, 1 from this, so it's become total 12 there. And then break into the two part, that is 50-50. So carbon, it's become C6, it become 12, it's become 6 there. So this is the case that when you to add the sucrose into the water, we are getting 2 unit of the C6H12 and O6. And what is the name here? One is glucose, one is fructose. Formulas will remain same for all the kind of monosaccharide, but that simple structure is different here. Okay, so I guess it's now clear. The first point, and the second one, we are using sucrose there. Okay, so the second method, yes, sucrose. So in this case, look at this. This is C6H10 and O5 is there. Again, it is a general formula, and if you to add the acid there, H positive ion. I said H positive ion. So what is happening then? Again, it is going to convert in the glucose. So important be like reagent. In this, we are using water. In this, we are adding diluted HCl. Okay, all the water in both the cases. But if you're using sucrose, glucose only, and if you're using, sorry, if you're using starch or cellulose, glucose only, and if you're using starch, so sucrose plus fructose. That's it. This is the only difference between both the cases. Okay, take some time, analyze the thing, and let me know if still not clear, so I can explain one more time. Actually, it's the same thing but with a different formula. Okay, is it clear now? Okay, good. So let's back toward the point structure, right? Okay, so this is how the chain, okay? Here we are having a two structure, that is open chain structure or a cyclic structure as well. So this is my open chain structure. Okay, so this is a open chain structure. It clearly observed here, that is a straight chain here. The point number one, open chain. Now, do you know that maybe everyone you know the small l and a small d there. So what is it? It's a dextrorotatory and it's a levorotatory. In term of the plane polarized light, if the plane rotate the light into the anti-clockwise direction, so it's become L, and if that is a clockwise direction, so it's become dextrorotatory, and it is represented by plus D and it is represented by minus L. The point number one. But do you know how to write the glucose? It's actually D glucose. So first of all, be noted the capital D and a small d is totally different. It's totally different phenomena. Be noted. Okay? So it's a totally different phenomena. What is capital D and what is small d? Small d is dextrorotatory, L is the levorotatory, but capital D is a different point here. Now for the capital D, what is capital D? Okay, it is a fixed thing. Okay, you have to check it for the last carbons, last carbon. If this kind of structure have to be there, okay, it is a CH2OH, okay, and OH and this, and it can be any R, okay, any 6 carbon, 7 carbon, 8 carbon, any number of carbon have to be there. If the last second carbon have this kind of configuration, that is our O is on the right side, okay, and H on the left side. So this kind of configuration known as a capital D, okay. And if the same thing, if the carbon, if let's suppose my OH, the last second carbon, I said last second carbon. So again, it become L. So glucose is a D type of configuration. So it's a L there. Okay, so let's just back here. If let's suppose sometime this problem has with the student be like, ma'am, it is very confusing. So let me just write it is a CH2OH. I guess it's sorted now. Okay, now we can simply go with the second fifth carbon or the last second carbon and observe this. Okay, noted this point here. It is actually the capital D. Okay, this is the point number one, capital D. Okay, so this is all for the normal general structure. So in the older time the glucose was discovered. Okay, so the first point, how many carbon present in a glucose? Now it's sorted for us, like we know there's a six carbon, but how to prove this there's a six carbon? So now we are going to prove every point one by one. That is a proof number one, six carbon. How to identify this? In this case, if you're observing there is a one aldehyde group, then how to identify? Okay, in this case, if you're observing total 5 OH group, noting 1 OH, 2 OH, 3 OH, 4, and total 5 OH group. One is a primary alcohol. Which one? The last one is the primary alcohol. Okay. So now our main agenda is going to prove the structure, justify the structure that 6 carbon, aldehyde, 5 OH, and 1 OH is there. Okay, so this is the next point. Anyway, let's just back toward the slide structure part where we're discussing. Yes, the slide done here. Look at the structure here. So let's read some theory about the structure. So what does this theory said? Glucose contains six carbon atom and one aldehyde group. That's why it's known as aldohexose. Aldo means aldehyde group, hexose means six carbon. Glucose is a monomer of a starch, glycogen, and cellulose. Okay, what is monomer? So actually the problem is what right now polymer chapter is related. So listen my point. That is everyone, I guess you know polythene. Okay, it's a simple thing. Polythene, at least you know the packet, the polythene packet we are using in our normal life. Okay. So what is actually the meaning of polythene? Poly is a so many ethene. Okay. So actually what is the case? If I'm using ethene, CH2, CH2, and there is a n number of ethene I'm using, and it will combine like carbon, 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 and up to so on. It's like so many CH2, CH2 group combined. But what is the repeated part here? The CH is a repeated part here. CH, CH, CH. So what is the smallest unit of the polythene? That is, you can say, ma'am, it is going to be the ethene. So what is monomer? That is a single unit. Single unit here. So glucose is the last form. We discussed glucose, fructose, and galactose are the smallest unit. So starch is a polysaccharide, cellulose is a polysaccharide, and our glucose is just a monomer. The smallest unit of the carbohydrate is glucose, fructose, or galactose. Okay, so this is written on the previous slide where we are. Okay, here, look at this. Okay, so glucose is a monomer of the glycogen and cellulose. What is glycogen? That is a stored sugar in our body. We discuss, right? Starch in plants and glycogen in human beings, and cellulose as well. And the most abundant organic compound on the earth is the glucose. Okay, okay, don't say, ma'am. Sometime you said hydrogen, sometime we be like oxygen. Okay, no, oxygen is an element in the universe. Hydrogen is the most abundant, but it's a compound, compound be like C6H12O6. you know every organism carry this. Not even that is the akaryotic, even eukaryotic, akaryotic, eukaryotic, everyone, smaller one, larger one, everyone. So it's really important. Okay, then that's it. Now let's typically talk about the glucose. That is, you can the structure elucidation of the glucose, or like in Hindi meaning that how to prove, okay? Like in normal language, that how to prove this. This is glucose. So molecular formula, presence of six carbon, presence of the CO group, presence of the CO as aldehyde, presence of 5 hydroxy group, presence of the primary alcoholic group, and the spatial arrangement. It is a very much important question in board exam. 5 mark question. Explain the structure of the glucose. Explain the open structure of the glucose. Explain how to prove, how to justify that carbon has a six, sorry, glucose has a six carbon. Okay, so let's discuss all the thing one by one. So first of all, let's talk about the molecular formula as we already covered, C6H12O6 is the formula for the glucose, right? And let me just give you some point. We just covered all the points, so I'm just giving you a brief. For assignment of the relative DLN configuration for monosaccharide, the lower asymmetric carbon we have to compare. Lower means the last second carbon, not the last one, because that was not asymmetric. Okay, I guess you know at least the basic rule of the, no, where was the structure? Okay, I guess you already know the basic rule of the asymmetric carbon, a carbon which is combined with four different group. So look at this. You know the what was the meaning of the last carbon? So see, in this case, this is not asymmetric carbon. It is combined with two hydrogen. This carbon is the lowest carbon which is combined with four different group. Now you'll be like, ma'am, how it is four different group? Look at this. This is the group number one, the complete giant chain. This is a group number two. This is a group number three, and this is a group number four. So it's be like you can say it's a chiral carbon. Okay. So everything has an importance. It's not like we're just using in a theory. Okay, why I'm explaining each and every point in very much detail, because the theory of this biomolecule chapter is not, good. So everything has a logic, everything has a point, and NCERT is not that much simple to understand for this chapter especially. So anyway, that where we are. Okay, let's back here. So if OH present on the right hand side, we discuss. If this side, right hand side, so it's become capital D. If left side, it becomes small d. D plus, D minus is the symbol independent upon the assignment. They just simply represent the dextrorotatory, levorotatory, just the direction of rotation. If plane polarized right is moving toward the anti-clockwise direction, so levorotatory. If clockwise, dextrorotatory. And the most abundant form of the glucose is the D. D means capital D means the last second carbon left hand side OH. Plus means dextrorated, it means clockwise direction rotation and glucose. So capital D plus means dextrorotatory glucose is the most abundant type of glucose present on a earth. Okay, that's it. This is a simple meaning of the assignment of configuration there. Okay, so shall we to continue with the structure, or I guess no, right? Because already so much time. So let's cover the six carbon and all these things, that how to prove the structure. Okay, that part in the next class. I guess enough, because so much theory I can understand your feeling. Okay, so that's it. So this portion, that how to elucidate the structure there, we will cover in the next class. Okay, so that's it. Next class will be starting from here. Now your turn. Any problem, any doubt? Okay, so you can ask. Yes, please. Your side now. I guess no, right? So I'm waiting for 1 minute if you want to ask something. Okay then. So how many session? Okay, Ambika, basically there's a three session, but according to my speed, because I have to finish at least this portion here, and it will take some time. So I can say four session at least. Okay. Although actually three session, but I have to explain something, then it will require four session at my end. So that what is the day? I guess Friday, right? Hmm. So Saturday, Sunday, Monday. So probably till Wednesday, okay? I will finish this. Okay then. So thank you so much then for your timing, for joining the class. Now you can leave. Thank you. Have a nice day.